Hey, Matty, what are we talking about today?
The Wuhan virus.
I don't think it's called that.
The novel coronavirus.
No, I don't think it's called that either.
SARS?
No.
COVID-19.
Well, I think that's the disease that it manifests. I think the virus itself is called SARS-CoV-2.
Okay.
But I don't think the WHO, not the band, I don't think the World Health Organization likes that name because it's too similar to SARS. So I think they just like to call it COVID-19, the disease, and the COVID-19 virus.
Oh, okay.
And that's what we're talking about today.
All right.
Welcome, everybody, to another episode of Dr. Matt and Dr. Mike's Medical Podcast. Today, we're talking about the coronavirus. Obviously, this is something on everybody's mind, and I think it's worth discussing some important points. Today, we're going to go...
We're in lockdown at the moment in my house.
Well, we're not actually in lockdown, but we are in Matt's
Could be
house.
by the end of the week.
Potentially. We'll see how things go. We're just going to see.
Which room do you want to be locked in?
Probably my room at my own home would be good, thanks. We're going to talk about brief background. So where and when it began. We're going to talk about what it is. So different types of coronaviruses, especially ones that are around currently, how it works, how it's transmitted from one person to another, what the symptoms are. We're going to compare it to the flu, SARS, and MERS. We're going to go through some stats. So current stats as of today, which is the 16th of March, 2020. We're going to talk about the interventions, quarantine, and testing. And then we're going to talk about what you can do, because the biggest way to change this is through you. All right? As a listener, as a person, we can change this and make it not as bad. So, Matt, let's talk about a brief background. So this, now I think we should just call it COVID-19.
That wasn't a cough. That
Sounded
was just
like
me clear in my throat. As a preface, I want to just say, at least from my behalf, I'm not a virologist or an infection control expert. So what we're talking about today is just kind of synthesizing information that's provided by health authorities and also journal articles.
That's right.
So
we've done our best.
Yes, that's right.
And we are, now it's bound to change. 16th of March is today. Stats are changing daily, and I'll tell you about some changes in stats shortly. So, you know, one of the best places to get your information would be from the WHO or your local health authority.
Yep.
Now, brief background, where and when it began. So as far as I'm aware, this particular strain of coronavirus began in China, specifically in an area called Wuhan, in 2019, at the end of 2019. Now, they stated December, but it seems like the evidence now states that the first case of pneumonia from the virus was in November.
Yeah.
So we think that it's coming from Wuhan. Now, whereabouts in Wuhan?
Well, I think Wuhan is the city.
Gotcha.
Could be wrong here, but I think Wuhan is the city, and it has it's a sizable city. But it sits in a province which I don't actually have on the top of my head.
Hubei.
There you go, Hubei.
Yep.
So it's thought that it originated in a wet market.
So a bunch of animals.
Yeah, we'll get to the different types of corona in a second. Is there anything else you wanted to talk about when it first sprang up?
Well, I just think it is important to say that it came from an animal to human.
Okay, all right. Well, we'll talk about corona then as a general term.
Yeah.
So corona just means crown. So this is a Latin term meaning crown. Now, it would be named accordingly to its morphology. So if you were to look at this virus under a electron microscope,
yeah,
you would see it has a fat envelope similar to our membrane, which is a fatty membrane, a fatty cover, and it is embedded with these kind of spikes which make it look like a crown.
And they're proteins.
And they're proteins. And so there's a whole family of coronaviruses. Now, they seem to have an animal reservoir, which are bats.
So all coronaviruses seem to sit within bats?
I believe so, as a reservoir.
So that means coronaviruses are what we call zoonotic. So
Yeah.
they can go from animal to human and vice versa. So basically can be transmitted between humans and animals. And like you said, beginning in bats, or maybe not beginning, but there is a reservoir, consistent reservoir in bats,
Yep.
but can be transmitted to other animals. For example, this current coronavirus.
Yeah. So the different coronaviruses that are known to infect humans, there's 4 different strains of the common cold.
Yes.
Okay. And they're named.
Which is a coronavirus.
Like alpha, maybe beta. And then there's 2 more newer forms of corona, and this was known as SARS.
Yep.
So severe acute, is it
Yep.
acute respiratory syndrome?
That's right.
That's what it kind of causes. And then there's the MERS, which is the Middle Eastern respiratory syndrome.
Correct.
Now, this is the newer one, so this is why it was initially called novel corona, but novel meaning new is probably a poor term because if another one comes, then it's not new anymore, right?
Yeah.
So they've changed the name to COVID-19, which is not really related to a people, a location, an animal. It's just the type of virus and when it first manifested.
That's right.
Hence the 19.
And the reason why they call it, SARS-CoV-2 is because genetically it looks like the first SARS virus.
Okay.
And CoV because it's coronavirus.
Yeah.
2 because it's the second type of the SARS. But a lot of authorities don't like this.
And the D was disease, right?
Yes.
Yeah.
And so a lot of people don't like this because it sort of insinuates that it is SARS, but it's not. It's something novel. It is something novel and new that we haven't seen before.
And so for SARS-1, it went from the reservoir, being the bats, into civets, which is a type of cat, then into humans.
Yeah.
For MERS, it went from the reservoir of bats into camels, then into humans.
Yep.
And then for this particular one, it's thought that it's gone from bats into
pangolins,
which is kind of like a scaly anteater.
Yep.
Into humans. And that was thought to at first originated at that wet market in Wuhan.
And we don't know whether it came from particulate drops from the respiratory system or through ingestion.
Yeah.
Because we have found viral particles within fecal material of people who don't manifest gastrointestinal symptoms. So we don't know how it moved through that way.
Yeah.
I suppose it doesn't really matter because we now know it is transmitted through droplets and particles and potentially through fecal oral transmission, which we'll get through shortly.
And I think the start of it in Wuhan was a direct transmission from the animal, and it took to a certain point until it became human.
Yeah.
Yeah.
All right. So how does it work? So we get this virus. Let's say we inhale this virus or we get some droplets into our nasal cavity or mouth. Where does it go? What's it want to do?
So very simply, the virus is a fat envelope, it's got a membrane around it similar to our membrane.
So it's just surrounded by fat.
Yeah.
Right.
It has these spikes in it, which are proteins. There's different types, and they do different things. But one of the most important one is an S protein, which seems to lock onto our respiratory cells. So when it comes down your respiratory tract.
So how far down? Because we've got the back of our throat, we've got our trachea, bronchi, bronchioles, and the actual tissue in our lungs. How far down?
Well, I'm not 100% sure, but it seems to bind onto a receptor called the ACE2 receptor, which stands for angiotensin converting enzyme 2
Which
receptor.
I think is
more lung.
Yeah, more lung.
So this is right at the bottom.
Yep.
But it wouldn't surprise me if it has the ability to lock into higher respiratory cells and then do its business and then drop down further.
Yeah.
So I wouldn't be surprised by that. But it seems that the S protein can lock into the ACE2 protein, and then once it does that, it kind of engages with our own cell membrane. Now, this type of virus is a RNA virus, so it has a strand of RNA inside it, which is its genetic material.
So all viruses are basically fatty envelopes filled with DNA, and they hide.
Genetic material. So DNA or RNA. I think it depends on the type of virus, but this one's RNA.
And they hijack our system just to make more copies of their DNA. They get our body to read their DNA and produce their proteins and produce more of their viruses to continue propagation.
Yeah.
So that's how they replicate, is by using us to make more of them.
Yeah.
So is this virus the same?
Yeah. So very simply, in our cells, we have our nucleus, which is filled with DNA, and along the DNA code, we have certain segments, which is called genes. And if we want to make a protein, give me an example of a protein we might want to make just normally.
We might want to make a transcription factor that reads more DNA.
Give something a bit more common that people would know.
Oh, okay. Let's say
a
protein to make your hair color black.
Okay.
Melanin.
All right. So you want to make that protein. Now, there's a section of your DNA that would need to get expressed to code for that. And so the DNA just splits apart. That part of the DNA is read, which is the gene. And when it reads it, it spits out a molecule called RNA.
Mm-hmm.
Now, the RNA moves, that particular RNA that's for that gene, moves out of the nucleus into your cytoplasm, let's just say, to make it simple. And then we have certain machinery, in this case, a ribosome, that reads that RNA, and then your protein's made.
Okay.
Okay?
And now you've got something functional.
Yeah, that's right. Now, what this virus does is it puts the RNA into your cell, and your body, so your cell can't distinguish that that's a foreign RNA molecule.
So it doesn't know whether you've made it or someone else has made it.
That's right.
And it wouldn't think something else has made it because it makes no sense.
So it just reads it
Makes
like
protein.
it normally would and makes a protein.
And that protein will assemble into the virus.
Wait, the protein it makes is what we call an RNA polymerase, which is an enzyme to keep reading that RNA molecule.
Ah, right. So it
So the first protein that it makes is an RNA polymerase.
So it's like making all the tools to build a house so it can just build more houses itself.
That's right. And so then that RNA polymerase just reads the RNA, but backwards, the opposite way.
Yep.
And when it does that, all it's doing is just manufacturing its own proteins.
Well, so if it's read one way, it makes the tools that can read it in the opposite way.
Correct.
That then makes more viruses.
Yes, that's right.
And then those viruses, what do they do?
And so really amazingly, the proteins that it makes, remember the ones that make the crown?
Yeah.
It just pumps those out.
Okay.
And all these just slot along that cell's membrane, your own cell's membrane.
Oh.
So now your cell is just filled on the outside with these
viral
spikes.
proteins.
And then at the same time, it just starts manufacturing it's the same RNA that it first puts in, so it just duplicates it, photocopies it. Copy. And as it's doing this, that RNA goes to the surface of your membrane and just pops off a new virus. And this just happens over and over and over and over again.
Wow.
So you started with one virus cell, and then just from that one cell that got taken over, millions are made.
And so does this then mean that, so then the cells...
And while it's doing this, before you experience a symptom, this is what we call the incubation period.
While it's producing these viruses.
Yeah.
Okay, so let's now say, for example...
But you are contagious at this point.
Okay.
Because as you're making this, you have the ability to then just expel it out your respiratory tract to anyone.
So the cells that line our respiratory system make mucus as well, which means that the mucus you produce in your respiratory tract will contain the virus.
Yes.
And if this is sneezed or coughed out, it contains the virus, which can then infect somebody else and do the exact same thing.
So if I cough all over you as I'm shedding, because this is called shedding, then you're got a huge likelihood of then getting that virus put into you. Whereas if I cough onto a surface, then if you somehow put your hand on that surface at a later date, then you could put it into your mouth or your eyes or your nose, and then it could infect you.
So that's the major method of transmission is coughing, sneezing, droplets of sputum basically.
Aerosol,
and also landing on objects, going onto your hand, touching somebody else, somebody touching objects that are contaminated with it.
Yeah.
So, and that's the primary transmission method.
And just, sorry, as a slight side point, it seemed that if you were to get inoculated, so these aerosols, polluted aerosols onto you, it appears, at least from the data in China, if you get a big inoculation, so a big load in one go, your likelihood of a more severe case is a lot higher. So all the doctors, all the health professionals in Wuhan at the time that got sneezed, coughed, spluttered on while they're trying to, let's say, put airways in or intubate or bag them, and they were just coughed upon, then they were more likely to get a severe form of it, opposed to if I just say coughed onto a surface and then you just introduced a few.
So viral load seems to matter.
Yeah.
Okay.
For severity at least.
All right. So when it comes to the symptoms then, so somebody has picked up it from the surface, been coughed on or whatever it may be, the symptoms seem to be that most individuals get a fever. I think up to 90% of people manifest with a fever.
And do you know how long we're looking before the symptoms come out?
Great point. So let's just say you touch a surface. It's got a virus on it. You put that surface into your mouth or touch a mucosal layer, nose, eyes, whatever it may be. It seems to be, one, the virus can last on shiny or metallic or smooth surfaces for up to 9 days.
That's probably best scenario.
That's best scenario for that virus, right? Worst scenario for us, but best scenario for that virus. But usually, I would say on average, a day under normal conditions. Once it's in your body, it seems to be about 5 days that it incubates before you manifest your symptoms.
So what I just explained, that's the incubation period from when the first cell hijacks to the point when the first symptom comes about.
That's right.
Okay.
Yeah, around about 5 days, but it can be.
But you are contagious.
Yes, so it can be up to 14 days before you manage your symptoms. One of the reasons why 14-day quarantine. But you can be contagious up to 48 hours prior to you presenting with symptoms.
Yeah.
So this is one of the reasons why when you have a look at people who have just been diagnosed, or they ask you, when have your symptoms started? And they tend to trace back, at least this is what they're doing where we are living in Queensland. They're tracing back 48 hours to people you may come in contact with and places that you may have been.
Okay.
Because of, what that means is you pick it up from a surface. You're probably not infectious for 2 days, but you are probably infectious for the next 48, 72 hours before you present with symptoms. Yeah. Then you have symptoms, which can last 2 weeks, right?
Mm-hmm.
Up to 20 days. And then after that, the idea is your viral shedding should diminish after those 2 weeks. However, there's some evidence that you can virally shed for up to a month.
Yeah, it's amazing.
Right? So you can virally shed basically from day 3 to day 30.
And just with that, I spoke to a clinician and they said, yes, by far the type of viral shedding will be from the respiratory tract. But like you said earlier, you can then infect your gastrointestinal tract.
Yeah.
And you can virally shed rectally.
Yes.
So this is where some people can get infected through fecal oral route, I guess.
Which is one of the reasons why, if you're sick and you don't know if you've got it or not, use one particular bathroom if you can that other people within your house can't use.
Yep.
All right. Now, if we compare this to other types of coronaviruses or even the flu, for example, I think it's worth doing, and it's not to diminish this. It's not to do direct comparisons of cases or fatality ratios or anything. It's about perspective, but it's also to highlight probably the severity of what we're approaching, right?
Yeah. And I think it's also just important that we only have to note here that we only have really 3 months of data. So this is changing literally every day, right?
Okay, so give us the stats for today, as of the 16th of March. Globally, how many people have been diagnosed? So this doesn't mean how many people have it. It's just how many people have been diagnosed through formal testing.
So we currently sit at 170,425.
Globally. And how many, so we don't talk about death rates. We talk about case fatality ratios because it's ever changing. So it's a ratio. So What is the how many people have died? And then we can talk about case fatality ratio.
6,506 individuals.
Okay. So what that brings it to, just going off those stats, is around about 3.7, 3.8% case fatality ratio. Now, last week when we were on ABC Radio, which is nearly 7 days ago, the stats were 109,577 cases.
So 61,000 more.
Yep. And 3,809 deaths.
So it's doubled.
Yes. And it's still around about the 3 to 3.5 case fatality ratio. It seems to be that that's where this CFR, case fatality ratio, sits for this disease, is around about 3.4%. So that's 3.4% of people statistically have passed away from infections.
Okay, so if you were infected with this particular virus, 3.4 persons out of 100 potentially will die from it.
That's right. Now, if we compare this to the flu from the last flu season, so there's a number of...
It's influenza.
It's
important to note here because a lot of people get this mixed up. Flu means influenza. It doesn't mean the common cold.
Oh, yeah. Common cold is a coronavirus.
Or it can be.
Yes, true. Influenza is not.
Yeah.
Now,
influenza is a serious type of virus, a seasonal virus.
That's right. And so the flu, every flu season, or let's just say last flu season, 650,000 people died from the flu worldwide.
Say that again?
650,000 people worldwide died last flu season, which is a lot, okay? But a lot of people are saying, well, that many people died and we didn't, we haven't done the same measures.
Yeah.
But now think about this. The flu, there's this infection rate, right, called the R0 or RO, which is basically on average, if you got it, in a perfect world where everyone is susceptible, how many people are you likely to give it to?
I'll say if you're infected, how many people would you pass it on to?
On average.
Okay.
And for the
this
flu...
This is the R0 or the R0.
Yeah, so for the flu, it's 1.3. But for the coronavirus, it's 2 to 2.5.
So for the flu, influenza?
1.3 people.
And for this one?
2 to 2.5.
Okay.
So you're likely to give it to twice as many people.
So basically what this R0 means, if it was below 1, that particular virus would just die out.
Yeah,
okay.
a given time.
If it's 1, it may survive, but it's not really increasing in number.
Yeah.
But if it's above a 1, as you're approaching 2, that's almost like it's doubling.
Yeah, so we've got 2 to 2.5 for this COVID-19. So considering 650,000 people died worldwide last flu season, and it has a R0 of 1.3, COVID is 2 to 2.5, so more infectious.
And I've got a couple of other ones here if you want me to say.
In a second.
Okay.
The case fatality ratio for the flu is 0.1%, and the case fatality ratio for COVID-19 is 3.4. So it's twice as infectious and 10 times more deadly.
Okay.
Which means that if we use the current numbers for the flu, 650,000, it is not outside of reason to suspect that millions of people this season will potentially die of COVID-19. This is why it's quite severe. We'll talk about risk populations shortly, but let's talk about some other diseases with R naught values. Yeah.
Okay. So you said the flu, which is 1.3. COVID-19, which is this one we're talking about, is 2 to 3.5.
Yep.
Okay.
So I said 2 to 2.5.
Yeah. SARS-1 was between 3 and 4.
Yep. So that was in 2003, I think.
Okay. Now, if you go to the Spanish flu, which this COVID-19 is being compared to at this early point,
okay,
that killed potentially up to 50 million people.
Wow.
Its R0 was between 1.4 and 2.8.
Okay.
Okay. Ebola was 2.
Yep.
Okay. Now, just for perspective, Ebola, its case fatality ratio was 60%.
Whoa, 60% of people, 6 out of 10 people died.
And then one of the highest infections with an R0 is measles, which is between 12 and 8. So that would suggest if you had measles, if you were working in a naive population, 12 to 18 people would get it from you.
Okay.
But luckily, the R0 can be heavily reduced by vaccination.
And we have vaccinations for measles and the flu.
That's right. And mitigating methods of transmission. So this is why we're starting to see countries and authorities do certain things like quarantining, removing mass gatherings and so forth. Yes.
So I think maybe are there any other stats you wanted to discuss, or can we start talking about interventions and quarantine and testing?
I think it's time. I'll just give in some different examples there.
Cool. So I think the take-home message here is that we're only in the first couple of months of this new virus. We don't have a vaccine for this new virus. It's spreading very quickly.
How long do you think it's predicted?
Yeah, probably
next year.
Best case, 12 to 18 months, end of next year, I would say. Because it obviously needs to be produced, needs to be tested, then it needs to be mass produced and then delivered. So I would say end of next year. The good thing is it seems to be that this virus doesn't mutate significantly. So whatever vaccine we produce should be stable enough to be able to knock at least a good majority of it on the head. Now people say, well, what about the flu vaccine? Flu vaccine is 70% effective, which is great, which is why we only have 650,000 people dying, right? And not higher. So again, without a vaccine, this current season, it can get pretty nasty. And this is why we now need to talk about interventions, quarantines, testings, and then we can talk about what you can do.
I think the one thing you forgot to mention, we did allude to it, were the symptoms.
Oh, yeah. So the symptoms include fever, which I said 90% of people tend to manifest. So that's a most common symptom. So obviously anything above...
And that's fairly non-specific.
Yes. So, above 37.5, you'd be thinking a fever consistently.
And that would probably depend on how you're taking the temperature, because there's difference between peripheral and core temperature, but that's a separate issue.
You usually know when you have a fever too.
Okay.
Cough, so a dry cough is quite common. Sore throat, less common. So the two most common is dry cough and fever.
Fatigue,
and
myalgia.
sore throat. All these can be present, but around about less than 50% of cases present with sore throat and fatigue and myalgia. So cough and fever are the two big ones. The other thing is, at the moment, at least in Australia, we're looking at people who have been in contact with people who've known to have it,
Yep.
Or people who've travelled overseas. But maybe if you're somewhere around the world where it is spread quite extensively, just the symptoms itself could potentially warrant you getting tested.
Yeah, I think either yesterday or Saturday I looked at Australian stats, and I think we were sitting at about 250 people had tested positive.
Yes.
About 138 to 140 had been in contact with people travelling.
Or had themselves travelled.
Yeah, and there was about 50 who
Unexplained.
transmission locally.
Okay.
So that's now suggesting that it's spreading in Australia.
Yeah.
And then 50 that were unknown.
Okay. All right.
So it's starting to suggest that it's taken hold.
Yeah, absolutely. Now, testing. So how do we test for this? What's the current testing methods?
Well, I would imagine the most specific test is the swab, the nasal or the oral swab, which what I'm guessing how it would work is essentially there would be almost like a cotton bud with a long stem.
Yep.
Or I guess that's correct. Put it up your nose or back of your mouth, take it out, and then they do a genetic test for the virus's genome. And if it's present there, it's suggestive that you are infected with that virus.
So taking a swab from deep within the nasal cavity or the back of the throat and a sputum test as well. So this is being able to produce some mucus from the lungs. Seems to be that doing both is going to be most beneficial to a positive test for sensitivity's sake. Seems to be that coughing something up is the best way. It's far more specific than getting it from the back of the throat, so the pharynx, nasopharynx area. They're the two ways that they usually do it.
Yep.
And they basically confirm one another.
So this is, as I said, the most specific. So this would be the most specific test, but the problem is, or at least what they demonstrated in China, if you're in the early phase of your infection, you may not be producing enough secretions for it to be sensitive enough.
Yeah, I think it's 60% sensitive for the cough sputum production and only 40 to 50% for the naso swab.
Yeah. So do you want me to talk about the most sensitive test?
Yeah.
So in China, at least, they did a study of at least 1,000 people, and they found that the most sensitive test, at least in the early phase, was actually a chest CT, so a CAT scan of the chest. And what that shows relatively early on in your infection period is you'll have lung changes, so certain changes on the image to suggest you are starting to produce pneumonia-like changes in your lung.
And this is what's important, because what ends up killing people is the pneumonia that they develop ultimately, because the pneumonia basically stops the lungs from being able to take on board oxygen and throw it into the bloodstream. So people, shortness of breath, right, is one of the common symptoms too. Fever, coughing, shortness of breath.
Dyspnea.
Yeah, dyspnea.
Which would be suggested a bit later, but yeah.
So people can't get enough oxygen, and then they need to get oxygen through intensive care units. And so the issue that's cropping up at the moment is the fact that too many people are coming in requiring oxygen through this method, and there's not enough intensive care units to satisfy the need. And so what that means is certain people don't get
the intervention
that's required and pass away.
Yeah.
And now if there's two individuals coming in, maybe a 70-year-old and a 40-year-old,
and this seems to be the case in Italy.
Potentially the 40-year-old would be triaged while the 40-year-old will be looked after, because it's not an easy decision, but it's a decision they need to make one of the two need to be looked at because they've got limited resources. And this seems to be one of the major reasons why countries that are inundated are suffering such high fatality rates because of this reason. This is why what we're trying to do now is you don't want this curve of infection to just spike up exponentially. You want to flatten the curve. That's what everyone keeps talking about, flatten the curve. And what that means is we've got to a lot of us are going to get this at some point. It's just statistically a lot of us are going to get this. We all don't want to get it now. There's talk about the UK saying, oh, we need herd immunity and we all need to get this virus now so we can become immune. But there's going to be 20% of us who need hospitalization or medical intervention. And that means that's going to significantly overburden the healthcare system and more people will die.
Yeah.
So if we all, if at some point, not all, we, good number of us get this over time, we don't overburden the healthcare system, the death case fatality ratio is low, we all get looked after, then we all develop the immunity. Now, hopefully we can get the vaccine before we need to get the disease to get the immunity. Vaccines are a way better way to develop immunity because who the hell wants to get sick and require hospital intervention?
Yeah, that's right.
So anyone saying, let me get sick now, ridiculous.
Yeah.
Absolutely stupid to say that. You should never want to get a particular disease or infection. We need to be vigilant and we need to flatten the curve. Now, the thing is, how do we flatten the curve?
So a good example, of, at least in the later stage, how China managed this, or the reports that were given from China, is the way they did this. Firstly, we know that one of the first signs is fever, so a temperature. So they do high amounts of random temperature checks in society.
Yeah.
And if you were shown to have a temperature above, let's just say 38 degrees, you were isolated. So you're pulled into a health facility where you were then screened for other possible more common ways that would cause a fever, like influenza and so forth. So you would do that. And if you were shown to be negative for that, you would then, your chest would be CT'd. And so
they rule out other things.
Yeah. And so after the chest CT, they would look to see if you had any changes on your CT scan.
Yeah, because like you were saying, the CT, the lung changes seem to come up pretty early.
Yep, that's right. And so now if you were positive for that CT result, then you would go to a separate kind of quarantine point and you would be then swabbed with the PCR.
Yeah.
Okay. Now, if you were positive for that, you were then taken to another separate quarantine, which may be like a hotel room.
Yeah.
And you would be kept in there potentially up to 14 days until you weren't infectious anymore. But if you need an health intervention, I guess you'd then go to
one of the
health facility
clinics that they produce. So,
and by doing this, you're mitigating, because as we said, potentially, maybe I'm going to guess a number, 20-30% people may have such mild symptoms they wouldn't even know they had it.
Potentially.
And so they're going back out into society and infecting others. And this is an example of where South Korea did really well, because they broadly screened. So when other countries had only done maybe a couple of hundred or a couple of thousand, not even a couple of thousand, maybe 1,000, they'd already done 200,000 tests.
And so they were able to hit on the head.
So they were able to see, well, big percentages are actually infected, but they wouldn't have known they had it. So
or at least not yet.
We can then isolate them and take them out of the community to stop further spread.
The hard thing is the kits required for testing is limited, obviously. And so the reason why, at least in the early stages for Australia, for example, they'll only test individuals who have been overseas in the last 14 days or have been in contact with somebody. And this isn't because they're being blasé, it's because they have limited tests. And we need to limit the use of the test and use it most wisely. I think if we had a good number of these kits available, would be testing as many people as possible. Now, obviously, South Korea was hit early, so probably was able to get its hands on.
Well, it's actually hit almost the same time as Italy.
Yeah.
And they paralleled for a long time.
Yeah.
But then one shot up and one kind of has flattened.
Yeah, so there's
like you spoke about. And that's a good example of what happens when you flatten the curve.
Yeah, exactly. And so you're looking at these peaks. So China is past its peak. So new cases per day, at least what they've stated coming out of China is, 5 new cases a day. They're closing down temporary hospitals that they've built because they've passed their peak. Now, Italy hasn't. Italy is still peaking at the moment. I think there was 2,500 new cases just in the last 24 hours, which is huge. America's still on the rise. Europe is still significantly on the rise, and Australia is in its early stages of rising up and will probably peak in the next few months, 1 to 3 months. Now, this is the thing. We can't emphasize more that every single person can actually do something to help. You can literally save lives right now by doing very simple things. And these very simple things are washing your hands with soapy water.
Why is that?
Because you brought up the fact that the virus is covered in fat, and fat hates soap, and it literally sheds that fatty layer off the virus. It is the best.
Or breaks it up.
It breaks it up. It's the best method we have for getting rid of it. Now, most of these types of diseases are spread from the hand to the mouth. Wash your hands consistently, before meals, after meals, before interactions, after interactions. I know that it's not always feasible, but you need to wash your hands with soapy water. It doesn't have to be, the 99.9% kill off bacteria soap. It just needs to be soapy water.
Wouldn't be helpful there, would it? So let's just say you don't have soapy water.
Yep.
Is there any other material or substance you can use?
Hand sanitizers are useful. They're not as good as soapy water. I'm going to say that. It needs to be a minimum of 60% alcohol to be useful.
Okay.
And we always put a little 5-cent piece or dime drop on our palm, but the WHO actually states you need a significant palm-sized bit and rubbed significantly through the hand. The problem is...
So your whole hand is saturated.
Whole hand is saturated with it, with the gel. And the thing is, it doesn't clean your hand. It can inactivate viruses on the hand, but nothing comes off, right? So it's not even as close to being as good with soapy water because the rubbing with the soapy water and then the rinsing under the water, warm water, better. The warmer the water, the better. If you've got dirty hands, your hands remain dirty with the hand sanitizer, right? But if it's the only thing you got, it's better than nothing. All right.
So basically, to reiterate, both for the soap and the sanitizer, a big part of it is not only the substance but the mechanical action of you rubbing your hand and touching every part of your hand, fingers, thumbs, nails.
Yes, clean under the nails, clean between the fingers, behind the hand, around the thumbs, and you need to do it for about 20 seconds minimum when you wash your hands. So sing happy birthday twice or just count to 20, right? And make sure that you do this. This is so, just doing this will literally save lives. I know it sounds silly, but if you wash your hands, let's say just wash your hands 3 times more often than you currently do, right? You got to do it.
Yeah. Now, and avoid touching the T-zone.
Ugh, what's that? I'm the... What?
You're the T-man.
Oh, that's right. What's the T-zone?
Oh, I think it's just between your eyes, nose, and mouth.
Yeah. Just try and... Look, it's nearly impossible. We touch our face minimum of 20 times an hour. So try not to touch your face. It's not going to happen. Be mindful of touching your face. But if you wash your hands, it's not going to be an issue. Now, if you need to take public transport, this is difficult because one of the other things you need to do is social distancing. Minimum of 1.5 meters.
I'm good with that.
Yes.
I don't have any friends.
No, that's right. So say something offensive to all your friends so they don't want to see you. Keep your distance. No large gatherings. No unnecessary meetings. Don't go to the football.
Well, in Australia, we're down to 500 max, right?
I still think that's ridiculous. I'm not going to wait for the government to tell me not to attend certain things because they're going to weigh up economy versus health. That's just a personal opinion, so I'm not going to wait. So, in my eyes, schools should be closed. Universities should be closed. I'm not...
You're doing your lecture online this week, yeah.
That's right. So why, if we can minimize the spread, my students can learn online.
Yep.
If you listen to this podcast, it demonstrates you're learning online. All right. So social distancing, washing hands. What else is there? Not touching face. They're the main things.
Okay. What do you do if you suspect you may be infected?
Okay. So if you think you've been infected and you tick those signs and symptoms, don't go out. Minimize contact with other people. call your local health authority
Yep.
or your local GP. Don't just go to the doctor. Call them up first and talk to them. They can either tell you what to do next,
Yep.
or they will direct you to another line that you can call. So there are various health lines that you can call up, and you'll talk to them about your symptoms and about your history. And if they think it's significant enough to move on to the next stage, they will tell you, and they'll either tell you to go to a particular location, or they will come to you depending on where you are located. So
I think in Queensland we have fever clinics now, which are just separate sections in the hospital that will just be focusing on this.
But you call prior.
Yep.
Call prior.
I think in Australia, or at least Queensland, it's 13 Health.
Yes, 13 Health is the Queensland number that we call, and you can just call and ask. At the moment, if you haven't been in contact with somebody that you know and you haven't travelled overseas, it's likely they won't test you, and they'll tell you to stay home and quarantine yourself. If your symptoms get worse, significantly worse, then that's when you start to go seek that medical help directly. But you can always call up and ask. So I think you will definitively, I looked at this is what I want to finalize with. There might be another point you want to finalize with, but I looked at a curve that an epidemiologist produced showing the current trajectory. And if we were to mitigate one case transmission through social distancing, it already started to flatten out the curve, right? So here in Australia and anywhere else, if you can work from home, work from home. If you don't need to go to school, don't go to school. If there's parties and gatherings, you apologize and not turn up. If you can do these things and keep your distance, wash your hands, don't touch your face, and so forth, you will save lives and you'll flatten the curve, which means your local healthcare system will be able to deal with it. And that's what we want. We're not going to stop this thing. We can mitigate it in the short term, and we can lessen its impact. We need to take control of the virus, not let the virus take control of us.
That's a good parting message.
Thanks, Matt.
So have we ticked off all the points that we said we're going to cover?
Yeah, background, what it is, how it works, transmissions, symptoms. We compared it to other viruses. We spoke about stats, intervention, quarantine, testing, and what you can do, which is the most important part.
It's ever changing. It's changing literally by the hour.
There is a great podcast, which we're going to plug, called CoronaCast from the Dr. Norman Swan,
ABC.
Australian, but they do a 10-minute update every day. So have a look at CoronaCast. Thanks, Matt.
Bye-bye.